Hello, this is Gina Piscatelli with a lecture on water and mixtures for Anatomy and Physiology 1 at Madison Area Technical College. This information can be found in your reading assignment in Chapter 2, and this slide tells you the page numbers where you can find the different topics. To review the information, look at study guide question number 5 for this first unit in lecture. This is the last lecture for week 1, by the way. And in it, I'm going to start with the chemistry of water. The chemistry of water is important to us because all of our cells exist in an aqueous environment. That means a water environment. Most of our body fluids are mixtures of solutes suspended in water. And in fact, up to 75% of our body weight is due to water. Ultimately, all the chemical reactions in our bodies, and therefore the physiology of a body, will take place in a water environment or an aqueous environment and depend on the characteristics of water. Before I start talking about the structure and properties of water, I'd like to review with you the two types of compounds that were introduced in the chemical bonding lecture presentation. First were ionic compounds that are formed by the attraction of two oppositely charged ions. Because there are opposite charges joined but separated at either end of the compound, ionic compounds are said to be polar. This is much the same as the way a magnet or a battery is considered polar. And the example of an ionic compound that we saw before is sodium chloride, also called ordinary table salt, where the positive sodium cation is on one end and the chloride ion is on the other. The second type of compound that we looked at were covalent compounds, and they were formed by the sharing of electrons. An electron pair between two atoms is really evenly distributed between the two covalently bonded atoms. This occurs because the two atoms attract the electrons with the same force, or equally. Put another way, the two atoms involved in a typical covalent bond is nonpolar because they have the same electronegativities. And an electronegativity is the tendency to attract electrons. I'll review that term again, electronegativity. But an example of a nonpolar covalent bond and compound that we looked at before was carbon dioxide. The bond between the carbon atom and the oxygen atom is considered covalent and nonpolar because both atoms equally attract the electron pair. Another example that you can see in this cartoon in the lower right is a nonpolar covalent bond between two hydrogen atoms. Those two hydrogen atoms are the same as far as properties go, and so they both attract the electrons with the same degree of force. So back to the term electronegativity. The definition is the tendency of an atom to attract electrons. And electronegativity is different in different elements. How are you going to know what the electronegativity is between two atoms? Well, we're only going to see a couple of examples in this course, but in general, electronegativity increases as you move up and to the right in the periodic table. So, for example, oxygen has a greater electronegativity than carbon because oxygen is to the right of carbon. Okay, that's not why, but that's what we can see on the periodic table, and we can use to predict the electronegativity difference. Oxygen also has a greater electronegativity than hydrogen, and that will play a role in water. So although I have introduced covalent compounds as generally nonpolar, there are some covalent compounds that actually have what we call polar covalent bonds. And this is because even though there's a shared electron pair, those electrons spend more time near an atom that has a higher electronegativity. It holds on to them tighter. So you can see in this cartoon, between the chloride and the hydrogen, that's a covalent bond. But the chlorine, or chloride, is hogging the electrons, or pulling harder on the electrons. It has a higher electronegativity. Therefore, there's a partial, sort of, negative charge on the chlorine, and a partial positive, or sort of positive charge on the hydrogen. We call these partial charges sometimes dipole moments, and they're usually symbolized by this delta symbol that you can see. And I'll point it out to you here. This symbol right here is a delta. And so you have a partial positive on the hydrogen and a partial negative on the chloride. So the electronegativity of the two atoms involved in a covalent bond will determine whether that covalent bond is polar or nonpolar. So for example, in this table, you can see that oxygen has a very high electronegativity compared to hydrogen. So the electrons will tend to be closer to the oxygen in an OH bond. But there's very little difference between the electronegativities of carbon and hydrogen. So a bond between carbon and hydrogen won't be polar. It will be considered nonpolar. So now we can look at water, and it's an example of a molecule or compound that has polar covalent bonds. These polar covalent bonds exist between the oxygen and the hydrogens of water. And I'm sure you know already that the molecular formula of water is H2O. Structurally, one oxygen is bound to two hydrogens, and so there's two covalent bonds. But they're polar because oxygen has a high electronegativity compared to the hydrogen. As a result, there's a partial negative charge on the oxygen and a partial positive charge on each of the hydrogens. And again, here is that delta symbol that indicates a partial charge. These polar covalent bonds and the fact that the electron pairs tend to aggregate towards the oxygen atom gives water a very specific three-dimensional shape, in which the two hydrogen atoms are located to one side of the oxygen. This is due to the opposite partial charges and to the electron cloud that surrounds the oxygen nucleus. pushing the hydrogens to one side. So that's why it's usually drawn kind of almost in a triangle-like thing. Because water molecules have these polar covalent bonds, they interact with each other in a very unique way. The interaction is called a hydrogen bond, and it occurs between the partial negative charge on the oxygen of one water molecule and the partial positive charge on the hydrogen of a different water molecule. So these partial positive and negative charges attract one another and form what's called a hydrogen bond. So to really understand hydrogen bonding, you need to look closely at the diagram and notice that the red oxygen of one water molecule is attracted to the blue hydrogen on a different water molecule. So these hydrogen bonds that form give water very special properties. And these properties are ideal for some functions within the human body. One property is that water is a good solvent. It dissolves other polar compounds like electrolytes, and it can organize nonpolar compounds like lipids and fats. Another property of water that's good in our body is that it acts as a good lubricant for tissues and cells. And lastly, it's good for temperature regulation. In the next few slides, I'm going to explain how these properties of water work and define some of the terms that you see on this slide. For example, a hydrophilic compound would like water, so it would also be considered polar because water is polar. Whereas a hydrophobic compound doesn't like water. So that means it would be different than water and it would be considered nonpolar, which I've already got written there. Some other terms on this slide that don't make much sense yet are adhesion, cohesion, vaporization, and heat capacity. And I'm going to explain these in relation to these three properties as we proceed through the end of the presentation. So first, let's just talk about why water is a good solvent. First, water is a good solvent because it can dissociate other polar compounds like salts, electrolytes. Some proteins and carbohydrates are also quite polar. Recall that sodium chloride is an ionic compound, so that makes it polar like water. And all polar compounds are considered hydrophilic because they can interact with the opposite charges of water. So for example, notice that the chlorine shown here of salt, which is an anion, is attracted to the positive hydrogens, or partially positive hydrogens, in water. And then the opposite would be true for sodium. You're not going to be expected to write out this level of detail, but it's kind of important to see why water would dissolve a polar compound. But why is water good for organizing nonpolar compounds? Why is that good? Or how does it work? Because water influences how nonpolar compounds are distributed within an aqueous mixture. So an example of a nonpolar compound with which you're already familiar is common vegetable oil or olive oil that you cook with. Imagine multiple oil droplets that are immersed in water, like you're boiling water for pasta or rice or something. Well, what usually happens if you just let the water and oil sit without boiling it? The droplets merge, right? So what's happening in this instance is that water is repelling the nonpolar molecules. And so the nonpolar molecules seek each other out. They're attracted to each other. So they want to go towards each other. And so they aggregate together. So what we say is that water is organizing the fat or the oil or the nonpolar molecule. Okay, the second feature that's advantageous to our bodies is that water is a good lubricant, and it's due mainly to its hydrogen bonds. So I'm going to talk about why it's a good lubricant, but let's start with the definition of cohesion versus adhesion. So cohesion is the tendency of molecules that are alike to cling to each other. So for example, polar and polar. Adhesion is the tendency of one substance to cling to a different substance. So water will adhere to other polar substances. In cohesion, what we see is that multiple water molecules are attracted to one another. So both of these characteristics, cohesion and adhesion, even if the difference doesn't make much sense to you, makes water an ideal lubricant for coating body cavities and organs in the body. So let me show you the difference between adhesion and cohesion first. So in this capillary action, this picture on the left, we can see the process of adhesion working. And what has occurred is that we have a beaker filled with water, and there are 3 capillary tubes, which actually are supposed to show you the same capillary tube over time. So we'll say 5 seconds, and then 10 seconds, and then 20 seconds maybe. Okay, so this is time. And the inside of the capillary tube is coated with a polar substance. So water is attracted, because it's polar, to anything else that's polar. And over time, it creeps up the capillary tube. So that's an example of adhesion, adhering, like adhering to like. Cohesion is really due to hydrogen bonding of multiple water molecules. So those hydrogen bonds create a surface tension, and they keep this insect afloat. So that's an example of cohesion. But what does that have to do with our bodies, really? Okay, so one example of how lubrication, basically through adhesion, benefits our body occurs in the membranes that line our chest cavity and the membranes that line our lungs. So in our chest cavity, we have a membrane here. It's labeled as the parietal pleura. And then surrounding our lungs, we have another membrane. In between those 2 membranes are water molecules, shown in this larger picture over here. Okay? So the water adheres to both of these membranes. When our chest wall expands, this pleural membrane also expands outward. And because the water links the 2 membranes, the membrane surrounding the lung will also move out, as the chest wall does. That aids in our lung expansion. So the point is that the lubrication between the 2 membranes helps with lung expansion. Okay, the last property of water that's really important in our body has to do with temperature regulation. And water is good at regulating our temperature because it's very thermally stable. It has thermal stability. That means that it retains heat very well. It has a very high heat capacity, meaning that the bonds don't break without a lot of excessive heat to break them apart. Okay? But on the flip side of that is that water can act as a coolant. Okay, so when water does break apart, a lot of heat has been expelled to do that. And when the water vaporizes, that heat escapes from the body. This is called a high heat of vaporization. So, water can retain heat that maintains our internal body temperature. But if our body temperature gets too high, water can break apart or vaporize, and the heat will be lost. The heat will go from being stored in the liquid water to a gas. So that's why water is so important to our body. Now let's look at where water is located and some mixtures that we find in the body, a little bit more tangible. There's a lot of mixtures in the human body present in all our body fluids. All the body fluids consists of solvents, water as the solvent with various chemicals and compounds suspended in the water. Some of these fluids include the vitreous humor, which is in the eye, and that gives our eyes ocular pressure. Cerebral spinal fluid surrounding the brain and the spinal cord. Blood, of course, that's a very common fluid that we're all aware of. Urine, we're also very aware of urine. But again, as I mentioned before, there's fluid surrounding the lungs, there's fluids surrounding the heart, and there's fluids inside digestive glands like gastric fluid in the stomach, lymphatic fluid in the lymphatic system. So there's even bile, which is produced by the liver. So there's a lot of different mixtures and body fluids in our in the human body. The types of mixtures can be categorized into three groups: solutions, colloids, and suspensions. And all three types of these mixtures exist in the human body. Now, what's the difference between the different types of mixtures? They are distinguished according to four characteristics, if you will. How big are the solutes? So the size of the solutes. Whether or not the mixture, when left standing, will separate so that the solute and the solvent become separate and noticeably visible. The third thing has to do with, it's very similar really, whether the entire mixture will pass through a membrane, whether the solute and the solvent can get through a membrane. And the fourth has to do with whether or not the mixture will scatter light. What we're going to deal with the most are the first two, the size of the solute particles and whether or not the solute and solvent separate. So let me give you some examples and tangibles here. So a solution has a solute that's very well dissolved. The particles are really small. But a suspension, the particles are bigger, and so they're not dissolved as well. So the size of the particles in the three different types of mixtures are from small to large, solution, colloid to suspension. You do not have to remember these numbers, nanometers at all. The second feature that's important has to do with how the mixture would separate if it was left alone standing at room temperature. Really, only the suspension will separate upon standing. Both solutions and colloids remain dissolved, if you will. They don't separate out. The other thing that's kind of important is, I guess, this ability to scatter light. And you can tell the difference between a solution and a colloid because a solution won't scatter light hardly at all, but a colloid will. So I'm going to give you tangible examples now. All three of these different types of mixtures are found in our blood. Our blood is made up of an aqueous fluid called plasma, in which there are various things suspended or dissolved. So, for example, we have gases in our plasma, like CO2. We have proteins, like antibodies. And then we have cells, like red blood cells and white blood cells. Each one of these different things are solutes found within the solvent water or plasma. CO2 is the smallest, proteins are slightly bigger, and of course blood cells are even larger. So CO2 within plasma, that is a mixture that is considered a solution because the CO2 remains dissolved. It doesn't scatter light. The particles are very small. Proteins, on the other hand, in plasma, form a mixture called a colloid. So if you were to shine light through plasma that just has proteins in it, it would be a little cloudy, it would scatter the light a little bit, but the proteins would remain dissolved, or I should say, I don't know what another word is besides suspended, but it wouldn't separate out upon standing. Blood cells are the largest, and because they're so large, they weigh more, they're kind of heavy, and when they're left when the blood is left at room temperature, these cells sink to the bottom of a collecting tube. So suspensions are mixtures in which the particles are large, the solute and the solvent separate upon standing. So that's the difference between the three different types of mixtures. And we'll talk about some of those in A&P1 and some in A&P2. Thank you very much. That's all for the water lecture.